you open your page, talking about the protein synthesis. And in protein synthesis, here we need talking about the enzymes that help to build protein. Why we need to help to build protein inside your body? We need to build protein inside your body to form your characters. Now we have all the information inside your chromosomes. I need to take it outside and use this structure to form your characters. Now again, if you look here, now this is the cell again. Now as you can see, the nucleus, it's made up of nuclear membrane. If you look at the nuclear membrane here, it's made up of a lot of holes. Why we have these holes in the nucleus? So anything inside it can go easily outside. Here we have the chromosome. First step of protein synthesis is DNA replication, as what explained it in the first video. After we did DNA replication, the cells need to take this information to form a specific kind of characters. So what kind of characters? Specific kind of gene. So we don't need to copy whole the structure. We know that in the chromosomes carrying all your characters. We need just one character for this cell. Example here, if this cell is your hair cell, your hair cells need to form a specific kind of protein called creatine. So do we need to know all the information inside the chromosomes? No. We will just take the part that has the structure of forming creatine, and this part called gene started from here until here. So what is the first step of forming protein? Is replication. The second step is forming RNA, and we explain the difference between DNA and RNA in other video. We need to do the first step is to separate the two helix away from each other. Who can remember which kind of enzyme that separate the helix away from each other? Good job, is the helicase. Now we need, after we separate the two helix away from each other, we need to build RNA. It's a copy part of DNA, as we explained, and we all know that the RNA made up of just one helix. So do we need to copy these two helix? No, we need to copy just one helix, maybe this one or maybe this one. How is coming to build it or copy it? By using enzyme called RNA polymerase. Now the RNA start to copy the part by adding nucleotide. These are the nucleotide. It's found all around the nucleic or the nucleus. So these are the nucleotide, phosphate, sugar, and nitrogenous base. Let's add nucleotide now. Look here. If we have in the basic helix TACG wherever, now we need to add, if we have T, which kind of nucleotide we'll add is A. Good job. If we have A, which nucleotide we add? T. No, we will add something called U, because we all know that the RNA cannot have T. We have something called U. So if we have here A, we will add U. Let's move C with G, C with G. Who can remember how many pounds between them? Between A and T, how many pounds? 2 or 3. You can ask and you can answer it. So after that, after we copy all this strain, and we will not copy whole the DNA, we'll copy just this gene that we need. The RNA is formed and spread away from the DNA. So the two enzymes that help to form RNA is called helicase and RNA polymerase. Helicase for separated, RNA for forming the or adding nucleotide. Now the RNA that made from or copied from the DNA, I call it mRNA, messenger RNA. So in messenger RNA you can find inside the nucleus, because I can copy it from the DNA and it can go outside. Yes, it can go to the cytoplasm. So if they ask you where we can find the messenger RNA, we can find it inside the nucleus or outside in the cytoplasm. The second step, the RNA will move outside the nucleus by helping of the holes that found inside the nuclear membrane. As you can see, this is the hole that helped the RNA to go outside, carrying the information, forming the protein. Now this RNA will go outside to the factory that help to form protein. What do you think? What's the factor that's forming protein? Good job, is the ribosome. Now the RNA will go and connect with the ribosome. Now we need to read these messages. We all know, as we explained in the other video, that we have something called tRNA that connected with what, class? Connected with tRNA. This is amino acid connected with tRNA, and we all know that how many amino acids we have? 20 amino acids that connect with tRNA that carrying the code. Wherever this code, you have different codes depending on the types of the amino acid. Now what the amino acid will do? We'll go and check. If we have here UCA, what do you think the code that it connect with? U with with A, C with G, and A with T. It will go and connected with the code that found in the RNA, like here, started to connect the codes together. Now we are copying the codes that found in the messenger RNA by tRNA. The tRNA connected with amino acid. The amino acids start to connect with each other depending on the code. If we have any problem here or in the gene, we have different kind of protein of amino acid here. This protein will get changed and you have problem in your hair. For example, if you are making creatine inside your hair, that's why your hair become not normal, a little bit bad. So because there is difference or problem in the gene. Now we have here amino acid, group of amino acid coiled together to form protein, specific kind of protein. Any change in amino acid or any change in the code, different protein will form, different structure will form. So take care about your body. Be sure that you are eating healthy food to make a nice and good protein inside your body. And thank you.